I'm Henrik. I'm a researcher at Molecular Precision Medicine at Uppsala University, but I'm also working at NGI, where I mainly evaluate new methods. But I'm also can join in consultation and do service in case needed. Briefly, I will talk about the workflow of making having a single cell sequencing project and the methods, which could be good, how you choose methods, but also how the data, based on the method you choose, your data will be looking different. Some general principles that are used. Maybe you will deep dive or even look further into think about could you tweak the methods available and experiment even further. Some common method I would a bit diver into them, diver, di deeper into them, and then give a summary. And also at the end, I will give some uh, overview of the services NGI can offer on this topic. So, why use single cell RNA-seq? So, there have been these types of technologies have been used, for example, in heterogeneity analysis and cell type identifications, where here to the right you can see an example of how different cell types of neurons, mainly then in the brain, has been found by using these technologies. Like creating big atlases has been big projects using these technologies. But also lineage tracing during the cellular states in differentiation and development. And this technology has been very proven useful. It can also be used to look at monoallelic gene expression and splicing patterns and immune profiling on the single cell level. The single cell workflow. So it starts with selecting what species are you working with? Are you working on humans? Do you have a cell line? Maybe you have a animal model, and then that will influence how you design your study. How can you prepare your sample, and how you collect the samples? Maybe you want to collect over time. Maybe you need to collect at a hospital that is far away from the site where you have your lab. So these are considerations you need to take into account when you plan your experiments. And that will probably, it can affect the method you can choose depending on the technologies. And you need to spend good amount of time of having a good tissue dissociation protocol because it's really key to have high quality cells to get high quality data. If you don't have that, you will not get the best possible outcome from your experiment. You can, if the cells are dying, you will see dying cells in your data. Probably you will filter them out, and that's not probably what you're aiming to look at in your project. You want to have the best viable cells possible. And a good advice is to look what has been used previously with your cell type, because this will depend on the cell type, how you, what method you apply for dissociation. So it's very good to check the literature, maybe also talk to colleagues, and you can also reach out to NDI. Even if we don't do that service, we have users that have used various different tissues, so we can uh, ask also them for advice and give you. So that's really good. And also the companies. So we use mostly kits from different companies, and they could also help to see what tissues have been verified with using their methods and not. And then sometimes you need to do an cell enrichment. Maybe you have a very tricky tissue that the length, if it's a lengthy protocol for the tissue dissociation, you might need to have a dead cell removal step to remove the dead cells to get better quality data. But this depends on, it's good to spend not only reading but also trying out these steps. so you're sure you have good. So you dissociate your step cells, you check the quality of how is the viability. So you don't expect to just plan your experiment, run, and think you can go straight to the single cell. Uh, it's worth to spend time on these before you actually go to the single cell method. And then you come to the single cell RNA-seq methods, and those could be droplet based, fact sorting, microwell based, and different. And I will talk more about these in the coming slides here. And once you have done the single cell library preparation, you will sequence, and most often it's short read sequences, but nowadays it also comes some long read options. So you choose what you do for sequencing, and you also need to decide how much should I sequence. Do I need very detailed sequencing to look at fine details of the gene expression, like really fine details? Then you need a deep sequencing, or if you have do immune profiling, you might need to do deep sequencing depending on the method. And or maybe you just want to see which cell types, and then maybe you don't need so deep sequencing. And then once you have sequenced, you do the computation analysis, and that's what you're talking mainly in this course, and also have the support for an NBIS if needed. And so, just to mention here, these parts, the NGI, is what could help you with to actually do the sequence, library preparations and sequencing. So NGI has services for that. And importantly, and also what Åsa mentioned, once you have your data, you might probably want to validate that with additional methods. So here, what SciLife Lab has also additional the spatial methods. For example, the Visium spatial transcriptomics is offered this as a part also from NGI, but we also have in situ sequencing or spatial proteomics method or small molecular fish. So apply or spatial mass spectrometry, but depending. Or maybe you found one favorite gene and you want to confirm that you could do it yourself in a lab. But it's important to validate the result you come with, find with a single cell RNA-seq. And I guess for this short mention history, now it feels I've been a long time in the field. I joined as 2017 was already some method had been just published, but it started off already 2009 with not so long after normal RNA-seq, but it was not such a big field but at that time. And the very first, there was manual cells picked in 2009, and the single cells were sequenced. Later, this was multiplexed, which was an important part to be able to scale up, because sequencing a single cell, of course, would not be feasible. Only go one by one, and it had some different technologies for this. But what is still used nowadays is for some method you fact sort into individual wells. So that's also the and then liquid handling robots could handle the fact sorted cells. So you don't need to do manual pipetting of 3 to 4 well plates, for example. That's how you could actually do this in a feasible manner. And then came the nanodroplets, which really scaled up the number of cells possible to process at once. So these were the drop-seq and in-drop here, which is commercialized by the 10x Genomics that have released very thoroughly and good tested product that is reliable and really scales up the number of cells you can process per sample. Has also developed some picowells where instead of using plates, you have a matrix with many more picowells where each well also gets a unique bead with a barcodes. So you can collect more cells than on a normal standard 384 well plate. So you can scale up the numbers that way. And I will also talk a little bit more in detail, but it's called in situ barcoding or split pool barcoding, where actually the cell compartment became the cell itself became a own compartment, and you could by splitting and pooling scale up the number of cells substantially. So in this graph, it's how many single cells were in each study over the time. So you can see it really increases that the number of cells increase a lot. And this is a log 10 scale, so it's really increased. But in common between these, this is a even older slide I started off with, but it's sometimes it can still be that you manually pipette. It nowadays it's because you first want to measure on these cells, which electrophysiology, then it's called patch-seq. So you can like measure the electrophysiological properties, and then you later also want to see the gene expression. So it's still sometimes used in some niche projects. Laser capture microdissection is, I would say, it's not so common anymore. But fact sorting, you fact sort into a plate, or you have the microdroplets that's the 10x is basing their methods on now. There was a company called Fluidigm. I think they are still alive, but that uh, platform is not commonly used anymore. But what they did was to isolate individual cell and could transfer into individual compartments that later could be picked out. Then there was the picowells I mentioned, and in situ barcoding, where each cell itself becomes a reaction chamber and where reagents can flow in and out of the cells. And the next step is you add a barcode to the cell in that compartment in these different methods. And that goes depending on the method with different ways. If you have them in like a fact sorted well, it's easy to see. You just add the barcode there. But at the microdroplets, you add a bead with barcodes which attach. But this is the way to later step. You could pool all of those barcoded cells and sequence them together. So here is also a bit old slide by now, but still I will point at some common feature between these different methods. So the first step here was the cell isolation. So either you have the drop-seq, or maybe you have a fact sorted your cell like in the smart-seq, and then you do a reverse transcription. So you want to capture the mRNA. So you have most cases an oligo-DT primer. Some also use, in addition to the oligo-DT, also a random hexamer to get internal um, binding. But you have the oligo-DT, and in that end of that, you have a PCR handle. So you add a PCR primer sequence. And then you do the reverse transcription. And, oh, sorry. Here you could also introduce a UMI, which I'm sure you will talk a lot more about. It's a unique molecular identifier. It's a way to label the individual mRNA molecules. So each of the mRNA will create one cDNA that has a unique, had random, a unique sequence that you later could deconvolve which mRNA molecule it came from. And then to be able to do a second strand synthesis, you add nowadays in most methods, you have a template switching oligo, a TSO, that allows you to add another PCR handle in the other end. So you could then have your index oligo here together with the indeed that oligo. You can create uh, an amplification in later stages. And the way this works is you have the RT enzyme. Actually, you choose an enzyme that adds additional C when it's done. So it has some making some errors, adding additional bases, but you use these additional bases to have this TSO that is complementary to this C that the RT enzyme did add. And that's how you could add then another PCR handle, or you could some technologies add the UMI in this end instead. And then the next step, you want to amplify this because to be able to read out from a single cell, you need to create more products. So you didn't see DNA amplification. And nowadays, I would say this would be by PCR. Some early methods used in vitro transcription instead, but PCR is the way most methods work today. And then you have a cDNA amplified cDNA library, and in many of the methods, or in some, you have a way here you could have a QC step to see did I create the cDNA library from my cells. And then once you have the cDNA library, you want to do a sequencing library. And here you could do a fragmentation, it's called. So you fragment and add barcodes. And commonly used is a favorite enzyme from the single cell field is called TN5. So it's actually both cuts and add barcodes at the same time. So that's the fragmentation. Now
I'm
we will
sorry.
take, yes,
So in the previous slide, you mentioned that the UMI is unique molecular identifier,
yes,
right? And with that, we can identify each RNA molecule.
yeah.
Does that work on principle of you just basically load the cell with a lot of random UMIs, and then all of them are unique, and then you hope that all of them will attach to RNA molecules, right? Or?
Yeah, so the UMI is a part of the sequence. So if we take this drop-seq, so you have your PCR, you have a cell, this barcode is a cell barcode, and then in this case, the UMI was a random 8 base pair. And the combination here of random 8 base pairs randomly will make each of them unique.
Mhm.
Uh, and if this attaches, you will get that unique. Uh, and then you have your cell barcode and the PCR handle.
Yes, but my question, what I'm trying to ask is, how do we know that? Because at this point, we're still labeling all of the molecules inside of a cell.
Yes,
So in order to achieve unique molecular identification, then you just load every cell with unique UMIs, a lot of different unique UMIs.
yes,
And then you do that for each of the cell over and over again.
yes.
Okay, I think I understand. Thank you.
So yeah, each cell would get different molecules of these barcodes.
Mhm.
And you have so many different unique uh, because of the 8 base pairs, and if they are all random, you get they they will become unique, each of them.
Yeah,
Yeah. So in
thank
that
you.
case, this was a 8 base pair, but nowadays they have become longer to be sure that they are unique. As the numbers
Mm.
of cells you capture scaled up, by increasing the lengths, you will make sure you still get unique molecular identifiers.
I guess my only confusion was how many actual unique UMIs can we get with eight base pairs? But I'm sure there's a calculation, and then we can capture up to X amount of unique molecules.
I can just add on to that in the processing pipeline, we look at the combination of a unique UMI that binds to a specific gene after doing the mapping of the read also. So the unique combination of these 8 random sequences to a specific gene occurring twice in this big set of random UMIs is highly unlikely.
Yeah. Okay.
So I think it's that combination with whatever maps into the gene also that makes it almost impossible to have 2 copies
I
of the
see.
same.
I see. Okay.
And you also have a cell barcode. So it should be on the same
Mm.
cell, on the same gene, as was said. Yeah.
Mhm. Cool. Thank you.
Yeah. So I will go into example of SmartCheck free. So one of the methods I've been working with as it came out. So it was developed in the lab of Rickard Sandberg at Karolinska. And quite soon after at the platform at NGI, we started to offer it as a service. And it works by isolation by fact sorting into a specific lysis buffer that already contains some of the reagents needed. Uh, and here, so the cell was isolated and lysed into in the buffer. So now the mRNA is already lysed and freely floating in each well here in a plate. And then you add the reverse transcription oligo-DT with a PCR handling primer. It has the TN5, the reverse transcription adds these additional Cs, and then the TSO is modified oligo with Gs, so the actually RNA oligo here, then connected a UMI, it had a tag, and actually it's a partial TN5 motif. So TN5, which is, I will mention later, I will mention this soon. And then you could amplify by this TNF5, partial TN5 motif and the PCR, you amplify the cDNA. Here you make a QC step looking at the cDNA profile of each of the cells. So here it amplified, reverse transcribed and amplified all the mRNA molecules of those cells. And for each well, you get a cDNA profile. But in reality, what we did measure the quantity of cDNA in the whole plate and picked a few to see at the profile to see that it's a high size, uh, high molecular size of the cDNAs. If it's low, then it's a sign it's degraded cDNA and wouldn't create good quality libraries. When we're happy with the QC, we went on, and here is how the library looks at the cDNA. So you actually get several amplicons from the same cDNA molecule or from the same mRNA in the cDNA. Then we have this favorite enzyme, TN5, that cuts and adds these labels. And then it gets more or less random it actually has a slight preference, but it's more or less randomly cuts up. These fragments will create uh, pieces of cDNA that once more it's amplified and then it could be sequenced. And this uh, tags added is actually the sequencing priming reads. So you could know, so the sequencer could read them out. And this is kind of a little bit special uh, case because you will have some that's called the 5' UMI reads, they would have the UMI sequence that was added in the processing with the TSO, while the internal reads, they will not have UMI reads. So you actually need to take that into account when you do the data analysis and choose if you're going to use all of the reads. Then you have the full length information from the sequencing, or you want to use the UMI to have actually be able to count the actual mRNA molecules.
I have a
Yes.
question regarding the first QC of the single cell RNA-seq. Like, do you select a size range for these mRNAs to say, like, this has passed the quality control, like between 1 and 10 kV or something?
Uh, so this could depend a little bit on cell type.
Mm.
So I don't have any strict, but I would normally say it should. Uh, I would prefer if it's above 200 base pairs up to somewhere. Trying to remember, is it 2, 3 kb? But I should also say this depends on the QC method you use. So bioanalyzer will give the appearance one appearance. If you use a fragment analyzer, it will look slightly different, or a tape station. So the appearance will look a bit different depending on the QC instrument you use. That's an experience I had myself where I thought I had degradation and then I realized I had a more sensitive QC method. So, but it's a good thing when you can run more than one sample and then you get an impression like, okay, my cells normally look like this. If I have only, but if you only have small fragments, then you will, that's most likely it's all degraded. Uh, and then it's not worth pursuing. Another thing is this also comes from previously the library preparation part was much, much more expensive. Now it has switched a bit. Maybe it's worth to sequence and check in the subset, but maybe not go for the full big project before you have a nice data. I got more questions, I see.
I have,
Did I answer your question?
yes, absolutely. Can I ask another question? A very quick one.
Yeah.
Like during a bulk RNA-seq preparation, you run before starting even with the library prep, you take a bit and you go for a RIN evaluation. Do we do that, or should we also do that for the single cell then, like take a pool of cells and then run, or is it not necessary? It's during the preparation and the cDNA preparation, and the first QC step is enough.
Uh, if you have the possibility, I think that is a good way.
Mm,
Uh, and then you have good quality RNA. So if you already do it, I think you should continue.
okay.
That's a good practice, unless you change a lot what you need to do to your library prep, because then anyway it's not useful. But if you start fresh, if you haven't done it before, but with the word of caution, because for some methods now they recommend that you do it if it's a new cell type to you. So
Yeah.
that could be an easy way to start, like a first step. But if you have a method where this QC is part of the method, it might be worth running here. So yeah, it depends a bit on what method you have chosen to do.
Okay.
And if you have the access, so if you could do this yourself or not. Yeah.
Thank you.
You had a question as well?
Yes, thanks. I was just wondering, what is exactly the benefit of this TN5 motifs?
So the TN5 motif in the 5' end of the SMART 3, it was a small trick to be sure that the TN5 cuts there and adds the tag there. because
The UMI.
it's to the left of the UMI. So because
So it adds the tag
it adds the
at
tag
that
at the 5'
place.
end. They want to be really sure that the 5' end gets that cut and gets tagged. Since that molecule stops there, it doesn't go
Mm-hmm.
on. If it's an internal, you have sequence that is 5' upstream of that. But here it's at the end of the molecule, and they wanted to be sure that the tag is there, gets added there.
And then maybe I don't understand what is the difference between the tag and the UMI. I understand the UMI, but what adds the tag to it that it's, yeah.
It is to be sure also that it's a way to say this is the 5' end.
Mm.
It's the 5' UMI. I think I'm not sure if Åsa can help further, or you need to read that publication for the SmartSeq free, like to they explain quite well in the publication as well.
Okay. Thank you.
And I should mention that the SmartSeq they had later on developed this even further it's called Express, and then they took away the QC step here and the intermediate in a way to speed up and reduce cost, because it's actually a little bit costly to do this step when you do it for many as the project scale up and you want to process many cells. Was there any more questions before I move forward? So the sequencing here of the SmartSeq, you got the 5' UMI reads and internal reads. And this could, for example, be used in looking at different cell types. So you actually could look at the isoforms in blood cells, immune cells, for example. That's one example where how it could be used. And also the isoform, since you have the full-length transcripts. Now we'll switch to one of the most commonly used methods, is 10x Genomics Chromium 3' method. So here, the cell suspension is loaded into an instrument called a Chromium instrument. So what this instrument does is it you have the cells that goes into an oil and create a suspension where there are mini droplets that are liquid droplets or aqueous droplets in the oil, and also they have capture beads coming into this stream. And the capture beads in the simplest configuration, you have the oligo-DT, they have the UMI, a cell barcode, and what they call a read one primer here. So these capture beads are introduced so in a way that each droplet has one bead, and then some of these droplets will also have cells. And in these droplets, you have the cell and the bead with a capture sequence, and the cell is lysed, and the mRNAs are captured by these sequences on the beads. The RT reaction is made with the polymerase, and then the emulsion is broken, and all cDNAs from all of the cells in this reaction are together. And you get each cDNA is labeled also then with the UMI and the cell barcode that was in the capture bead. So each bead has a unique cell barcode here. And later this has evolved so one thing is each of these reaction, they charge quite a lot of money for buying these kits. So you might want to combine more than one sample per reaction. And then you could do cell multiplexing, and there are different ways to do it. But as one way to do is you have an antibody with a conjugated barcode. Then you take the same antibody, but you conjugate another barcode to this. That is then you incubate your each sample with each one of these barcodes. You pool them together, and now on the cell you have a different barcode on the different cells, depending on which sample they came from. So you have sample one and sample two. You get the sample one got the blue barcodes, sample two got the red barcodes, and then as you introduce them, the sample two was this example got you have these sample tag as well, and then you capture these in the droplet, and you break as you have amplified, you have a barcode library that is actually shorter in size. You could sort that out to a separate library, and then you have your gene expression library, and that is what you will sequence for the gene expression. But feature barcode this is called. There you could sort out which sample it came from, and it's the same principle for the site-seq, where you can actually look at the cell surface expression of different proteins, and that goes to a feature barcode library. And so this was the antibody conjugate with a barcode. There are other ways to multiplex. You can have a liquid conjugate that goes just to the cell membrane, not by antibody. So that's what 10x themselves call cells as cellplex. And another way to use this technology, you have the CRISPR guides that you have introduced to your cells, and those could be captured by also the feature barcode. And the 10x they developed, so not only do they have the oligo DT to capture the mRNA molecules, you also have two different capture sequences and later iterations of their kits. But you had a question.
Yes, thank you, Henrik. So I'm just wondering how many antibodies we can use for the multiplexing.
That's a good question. I don't know for this one, but I know. No, I shouldn't say too much. I don't know how many combined. Maybe.
I mean, it's all one antibody with multiple barcodes. So in theory, it's limitless because it's just a unique barcode on the same antibody. But I think the kits they sell for this, I think there's 96 plex, right?
Yes,
so,
because I remember, I'm sorry, I'm remembering actually from some of their presentations that they have limit on the multiplex that can be done based on the antibody conjugations. So, for example, I can't multiplex 96 samples with different, and no, I think it's different.
Oh, you're thinking about the ADT when you're doing detection of different proteins with antibodies, perhaps?
No, I think for the antibodies that we can use right now for the multiplexing, because the point here is that if I'm multiplexing, let's say, two different cell types based on some surface markers, because that can also be used, right? So if I have a couple of antibodies and each one has a unique UMI for them, then they can be multiplexed together.
Yes, but we wouldn't call that multiplexing in that. It's more detection of the cell types, and that's the ADT surrogate you're thinking about then. That basically you can tag different, especially immune cell, like all these receptors of immune cells, you can tag them in parallel to doing the transcriptome. And there's a limit in their kits what they have, but it's also a crowding issue.
Yes.
If you have too many different antibodies, I guess. Do you know more, Henrik?
Unfortunately, I don't have experience from that, so I don't know but I could check this up and I'll post in the lecture channel afterwards.
Thank you.
If I understand correctly, if we use this multiplexing with antibodies, that means that at the end of the day, if we don't put more beads, then we will get a lower sequencing depth per sample, right?
I mean, it all depends. If you put more cells in one run of 10x, you need to sequence more. So you need to sequence deeper. I mean, it's always the trade-off of amount of cells versus sequencing depth to how many reads you get per cell that needs to be optimized. Did that answer your question?
Yeah, I think so. Yeah.
But of course, I mean, if you load too many cells, there's also a risk of having more doublets.
I was just gonna mention that, but
Sorry.
I'm not talking so much about doublets, but it's the risk. So I mentioned that you also have empty droplets where you don't have cells. But of course, you want to use the capacity of the kit. Nowadays, they have, with the new version, they say you capture up to 20,000 cells per reaction. But at that rate, I think you have 10% doublet rate or something like that. And you will, I think, learn some tools here, but those are sequencing reads not useful. And you need to take that into account. And I'm not sure how efficient all of these tools are, because if you have two cells of the same cell type, it's maybe not so too trivial to actually decide if it's a doublet or not. So sometimes my experience is also this. These companies, they want to sell their kits saying we have a high capacity, but maybe they are pushing it the far to make the PI say, "Oh, it's not too expensive." But I normally we don't recommend to go what they claim is the full capacity to actually to have a trade-off between the doublets and to get better quality data, because at the end of the day, that our experience is much more valuable. So this was one cell multiplexing, and there was a paper published last year comparing different multiplexing technologies for single cell. So I could recommend if you want to do it, and I will mention one more paper soon. But I wanted to go through also the split pool sequencing technologies. So in situ barcoding, it was another name. So here you load your cell suspension, which each is a mixture of cells, but you could load one sample to each well here, or you could split them to more than one well. So in this example, it's the first row of this plate each well has a unique sequence. And then you do the RT reaction and a ligation that will add a barcode from that was on this well. And then you pull all of these wells, the cells from all of the wells, and you distribute them to a new plate where there are 96 new barcodes. And then you ligate that barcode to your cDNA. So you're still at the cDNA stage here, like you have one molecule. And then you pull it again and split it to the third plate with yet another 96 unique barcodes. And then you pull that again, and you have sequencing libraries, and they actually split these to different sequencing libraries. So you have finally 4 rounds of splitting and pooling that creates combination. And I mentioned before that here you have each cell is permeable, so you could add the reagents, but they won't go out. The mRNA won't go out of the cell, so it's contained within the cell. So you do these, and here is a calculation example from this example. Where you had 12 barcodes in the first round, 96 times 96, and you split this into 2 sequencing libraries. Then you had 221,000 possible combinations. And this configuration of the kit, this is what parts call a mini kit. So it's commercialized by parse this variety, and then they say you can do up to 10,000 cells with a low doublet rate because you had 221,000 combination. Then they have what they call a whole transcriptome kit. Then the first step was not 12, but half a plate, so 48 wells. And at the last stage, then you split that into 8 libraries. And by that, they say it's safe to collect 100,000 cells instead because they will get unique combinations. Or you go to the full plate in the initial step, and I think then you probably split also one more level at the last stage, and then you can collect 1 million cells. So this is the split pool. So it parses one technology. The other technology is called scale or commercialized by scale now. So they were two papers when they originated. It's also called SCI-seq, SCI. So based on these technologies with split pooling. And actually this year was a comparison of current method where they inputted a PBMC using all commercially available, or not all, but many of the commercially available methods, and they evaluated the quality performance, so analytical performance, and also experimental, like practical performances, how many cells they could get, and the cost, which is unfortunately a big factor when you decide. You need to take that into account. Both cost per cell, but also cost if you need to buy equipment at a time, and how long time it takes, the protocol time. And so they compared the 10x methods. I have not mentioned the one called here. They call FRP or Flex is the commercial name. So it's actually a probe-based method. So, well, I will come to that in the next slide. I see now. I will mention it as well. So that had a very nice performance. It's actually not sequencing, but a probe-based method, but with a sequencing readout. So quality-wise, it's very good. It's quite a high cost still. And yeah. So, but I really recommend to read this paper if you're going to choose method. And then I will have mention one more comparison, which I actually performed myself. So we have a drug screening project. So we compared the split pool with a parse, and we had multi-seq. So we used these oligos with lipid moiety for multiplexing, or we used the 10x fixed RNA profiling, which is probe hybridizing. So probes against all the genes in the 18,000 genes. They have designed probes for the human or mouse. You can use these. So they hybridize each, and they actually also add a sample barcode in the probes. So you could multiplex up to 16 samples in each reaction here. So we did compare the performance, and we also found that quality-wise, you got very nice reads with a probe-based method. The cost is fairly high. But I should say, I did this, we used the first version of the parse kit. They have since evolved, and probably they claim they have become better. So we are testing the new version now. So I just mentioned another thing you could measure beyond the transcriptomic is open chromatin, but you will hear more a later lecture in this course, so I will not talk about that. So a quick summary. So to make the single cell RNA sequence, you isolate the cells into a compartment. You add a UMI and barcode for a later pooling. Then you amplify your cDNA to be able to get sufficient product to actually read it with the sequencing. Defragmentation is often with a TM5 fragmentation, and you sequence your pool library and analyze your data. And then if anybody has questions so far, before I move to the next phase. Any more questions? Okay, then I move with the last 5 minutes here or so. I will talk a little bit about NGI. So NGI is a National Genomics Infrastructure. It's a sequencing platform. It started off, it actually offered sequencing services before SciLife Lab started, but now it's part of the SciLife Lab. It has two nodes, so it's both in Stockholm and in Uppsala, or actually it's three nodes. So here it's getting complicated already. So in Stockholm there is one. In Uppsala we have Uppsala Genome Center that makes long read sequencing, and we have NGI Uppsala SNP-seq technology platform for the short read services. But the NEI as a whole, we could offer sequencing with different sequencing instruments like Oxford Nanopore, PacBio. These are the two long read platforms mainly used. And then we have Illumina sequencers, different models, mostly the large size, but we also have some mid-size sequencers and a small. And then we have Aviti from Elements, that's a mid-size sequencer. And we can offer SNP genotyping. And we have different applications you can work, and also offer the spatial transcriptomics 10x Visium within NGI and OLIC Explorer. And single cell services. And I will focus a little bit more with the NGI single cell services. We do gene expression with the full 10x portfolio. We could offer anything kit they are selling. We could offer optionally also do long read sequencing of the 10x libraries with Oxford Nanopore. Recently, we had SmartCheck 3 that has recently been phased out. So the inventors, they also created this gene SmartCheck Express and has this as a service company in Stockholm. So they are very efficient, and we shouldn't be a competitor to a commercial company. So reach out to them if you want to do SmartCheck. We could offer parse or scale, so we'll discuss with that which ones. We are also currently evaluating ourselves internally, but we could offer it as a service if needed. The 10x genomic, the ATAC-seq could also be offered, or 10x ATAC plus gene expression, so that's going to be offered. Here in Uppsala, there has been developed a single cell SPLiT that looks at methylation pattern on single cell that could be offered in collaboration if needed. And recently also looking at the single cell whole genome, so DNA sequencing, that is tested some projects on that term. So we are happy to, if anybody is interested. And how it works to use the service is you contact NGI, we set up a meeting and discuss exactly what's your need, and we guide you what's the better method. Maybe you already know. If not, we talk what could be good. And we can also consult. You can use us for consultation. It doesn't need to be that we should do it. We could give you advice that maybe your project fits this better that you could do in your own lab. So we are happy to help you there. So reach out to the coordinators will guide you. And also we do the sequencing, and for the single cell methods, in most cases, if it's a common genome, we will do the mapping like run Cell Ranger or the equivalent. Yes.
Yes, thank you. I'm just asking about the service available, let's say in Uppsala. Do you have the bioscience sequencing option available?
I'm currently evaluating the last iteration of their kit. the service of parse that's being offered. So most of the service has been transferred. It's offered in Stockholm, but with parse you fix the cells. So that is you ship the cells to Stockholm instead, and they will help you process it. So we're really happy to discuss. But if needed to have live cells, we have the possibility also to run in Uppsala. And actually, maybe the next slide is kind of going there.
Thank you.
Very, very small commercial for NGI just also launched what we call an open lab. So at the platform, this is mostly for the sequencing part, it's offered at large scale. But maybe you want to tweak yours. Now I'm not only talking about single cell, but the library preparation method, and you want to do some specialized that could not be run. Since the platform is going high throughput, that's not possible to do tweak for individual users. But then you could book a time in our open lab, and as an expert from NGI could come and help you there. Exactly for the single cell part here in Uppsala. So we have opened this in Uppsala. It's planned to open also. The pilot platform is here in Uppsala. It's also planned in Stockholm, but their space is needed to actually be able to do it. But the idea is, if these seem, we are super curious about this, and we hope also to spread it in different parts of Sweden. But this could be a way to actually do it in Uppsala if you want to do it, but with support. But the premises needs to be thought through, because at the current lab we can't take live cells. But as I said, if you have a project request, you could reach out to this support email, or you go to the website of NGI and book a project there. You can book a meeting request, or if you already know what you want, you go to this link at NGI Sweden. And if you had any question, this is my contact email as well.